to the Weekly Bioanalysis, a KCAS podcast.
Hello and welcome to the 56th episode of the Weekly Bioanalysis, the official podcast of KCAS Bioanalytical and Biomarker services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. My name is John Perkins and I've been in the bioanalytical LCMS space for over 27 years. I'm here with my co-host Dominic Guarino.
Hello, my name is Dominic Guarino and I have been in the bioanalytical ligand binding assay and biomarker space for over 28 years.
Dom and I are senior scientific advisors at KCAS, and either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS's services. Today we're here with a special guest, Jeff Hester, Senior Director of Cell and Gene Therapy at KCAS.
Well, thank you, gentlemen. It's a pleasure. Yes, I've been, I guess, in the cell and gene therapy space, if you will, for several decades now. I'm showing my age, but I think that's a good thing in this space where expertise in various techniques and technologies are needed. I've been with KCAS a little over a year, but once again, been doing this for 20 some years. So thank you very much.
I'm located here in upstate New York. Dom and Jeff are in Kansas, and our producer is in Missouri, so we're all over the map. We are ecstatic to have you listening to the 56th episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcast, you can now find the Weekly Bioanalysis. We welcome all of you, whether you are joining us for the first time or if you're a regular listener. We started the podcast when COVID restrictions prevented us from continuing business as usual. The podcast was an idea we'd always had, but it suddenly became the perfect medium for us to employ. Actually, what we find now is we're starting to get out and talking to colleagues face to face, but we're still going to keep the podcast going. Today's podcast will be a review of the latest news and resources, and then a focus on a topic of our choosing before discussing any feedback we've had from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. Then we'll wrap up and give you a little teaser for next week, let you know what is coming in the future. So again, we're fired up to have you here, and we're looking for a fun episode today. To kick us off for podcast number 56, Dominic is going to cover today's podcast topic a bit before we jump into the news and resources section. Dom.
John, I love your energy today. You're all fired up and ecstatic. It's great to see you.
I couldn't be more ecstatic because I'm feeling almost normal after 2 weeks of being totally out of it.
Yeah, I could tell your energy's up. We love it. We're thrilled to have Jeff Hester here with us. We're going to talk about cell and gene therapies. I'm going to quickly go over the agenda with you right now. We're going to just, as I mentioned, we're going to talk what are cell therapies, what are gene therapies, what's the combination of them. We're going to talk a little bit about why it's exploding, how it's really like personalized medicine and how it's overtaken that. Then we're going to dive into some of the challenges. Then we're going to talk a little bit about the specific services that KCAS offers for cell and gene therapies. So with that, John, let's just jump right into the news. As always, we'll start with some COVID topics. And John, this is one I think that's been near and dear to us from certainly since the inception of the pandemic. And it's a story about how many COVID cases, how many COVID cases vaccines have prevented, how many deaths. That's what this story is about. So John, maybe tell us a little bit about it, but something we've talked about since the inception of our podcast. And certainly we feel like vaccines have been an incredible advancement in terms of preventing deaths due to COVID.
Well, I mean, sorry, excuse the voice. Talking as someone who's just been through COVID, etc., I have to emphasize how grateful I am I was vaccinated, because both my wife and I were relatively sick for a few days. But, you sort of think if we hadn't been vaccinated, how much worse could that have been? Because, for a few days, it really wasn't great at all. But this is a study by authors from the National Committee for Quality Assurance, the Commonwealth Fund, Yale School of Public Health, and York University, who've done an analysis of, they looked at data that looks at the national population demographics, cases, hospitalizations, deaths, daily vaccinations, vaccine effectiveness, and waning immunity, and then calculated from that how many deaths the vaccinations have prevented, and then the number of cases that, I think it really it's as our recent experience emphasizes, it's the hospitalizations it reduces. By their calculations from December 2020 when the vaccines were first authorised through to March 2022, their calculations estimate roughly 2.3 million deaths, and 66.2 million infections have been averted. And in terms of, it's not all about money, but they also calculated it prevented 17 million hospitalizations, to save in this country 900 million in spending on healthcare. So we obviously know from experience. You read the papers. There's less. The hospitalizations aren't at the level they were. So the biggest weapon we have right now are vaccines. Although both my wife and I, in terms of helping deal with the infection when we had it, we both had the most recent Lilly antibody as well, because they're old. We're both over 50. The doctor advised that we did that too. So there's obviously other things, but obviously vaccines have been a big success in dealing with this.
So you want to recap some of what you said. It was the Yale School of Public Health and York University who did the study, two real reputable places, right? They're estimating averted 2.3 million deaths. That's wonderful, and 66.2 million additional infections. That's amazing numbers there. And then 17 million hospitalizations prevented in the U.S. Close to $1 billion, 900 million in savings, but that's less of it. Those are. I think all along that seems right to me. This is so near and dear to you because of. Then you shared that you just got the infection. Then you went ahead and got some antibodies, the Lilly therapeutic. I almost want to ask you how that was. You can share if you liked it. I think a lot of us would be wondering, none of us, I've never taken a biologic. I've been studying them for decades, but never really had one injected into me. And you got a biologic, and that's, did it hurt? Did it do anything? Those are some of the questions I might have. If you want to share them, you can, but great story.
I'm happy to talk about it. I mean, the nurses knew my background, so one of them actually gave me the leaflet that comes with the treatment, so I could look at it. I can't quite find it right now. So it was I should have noted the name, but
Advavirem, right? Isn't that sorry?
There's two. So because of Omicron, etc., the older antibodies that we have talked about, they've basically been rendered ineffective. So now we're down to two different treatments. One is from Lilly and one's from AstraZeneca. I had the Lilly one because as soon as I was there, I was going, which one is it? Which one is it? And honestly, I think part of this one, the antibody treatment itself was probably about 5 minutes. So they just basically inserted the needle into a vein in your hand. They did the infusion for 5 minutes. It was rapid.
But it's a bag, right? This is an infusion bag, right? It's not an injection.
It was actually it was a fairly, but they were pressing it in, so they were actually speeding it into your hand, so it was a fairly quick one. So it wasn't.
But it's not like a needle, right? Just paint a picture. I'm just curious, right? It wasn't a needle.
To get it into your vein, they had. I mean, it's whatever you call the means of getting it into the back of your hand.
Yeah, so it's an infusion, right? There's there's a little bag on there, and then they're pressing that bag, and it takes about 5 minutes. Now, during that, do you tingle at all, or does it hurt at all?
I honestly felt nothing. It was just a case of do that for an hour. Sorry, do that for 5 minutes, and then they keep you for an hour to observe to make sure there's no side effects or anything like that. And essentially, it was trouble free. I mean, it was fine. It was, neither of us saw any issues with that at all. And so it was just like I say, one more.
Can't you jazz up your story? It doesn't have enough to it. It just sounds. No, I'm just kidding, John. That's wonderful. Then.
I will talk about the interesting piece because it touches on a little bit later. So my wife actually was the first one to get infected, and so she felt really ill the first day. And I was we were talking. I was looking at something on Twitter, and someone posted, "Can't believe it. The doctor won't prescribe Paxlovid via a telemedicine appointment, so they want an in-person appointment to get a COVID treatment." And I thought, oh, ding, the bell went off my head, and I said to my wife, "We need to call the doctor and see if you can get Paxlovid to just help you." So she was feeling so rough. I was the one who made the appointment, etc. But when it came to a questionnaire with the doctor, she had to step up and do it. But what I hadn't realised was that they're actually quite careful about who they'll give Paxlovid to because of potential drug interactions. So in both our cases, atorvastatin is one that is an interactor with Paxlovid, and my wife's also on an asthma medication. They're concerned about the possible effect of steroids as well. So that was why she didn't take it any further, and that was why she went for the infusion. When it came to me a couple of days later, my infection took a different route. The first 2 days, I felt absolutely great. I knew I tested positive, had no problems at all. And then on like the night of the second day, going into Sunday morning, I got this really extreme sore throat. And the next 3 days were really miserable, and you just felt terrible from that, the sore throat and other things. So I then did the same thing. Can I get Paxlovid? I think I called before things had got bad. I just wanted to deal with it quickly. And so talking to the hospital, and they're like, well, we can do you Paxlovid, but we have to talk to your doctor. And I'm thinking, I could happily drop atorvastatin for a few days, because then I can eat grapefruit for a few days, because that's a interactor as well. So I haven't eaten grapefruit in years, and it's like I was thinking, oh, grapefruit for breakfast would be fantastic. So I've got this all in my mind. I'm straight down to the supermarket to get grapefruit, and then I got a call late on the Friday afternoon to say, nah, your doctor doesn't want you to drop the atorvastatin. You're going for the antibody next week. So it was like, oh, like after that, it was fine.
Hilarious. But I'm glad you and your wife are well, and you felt like, I mean, I'm going to ask you a question. It's hard to answer, but you think those monoclonals helped, right? Obviously, like after you took it, did you feel better? Did it,
I honestly don't know if you detect whether it makes you feel better or not. I don't. I think I tested negative around. I tested on day 9 and I was completely negative by then. So I did. I assume it helps. I just don't know. I couldn't say my body reacted a certain way. Say it's the antibody doing this. I just honestly had no idea. But
maybe I would have willed myself with it. Oh, it's working. But no,
I had 3 really bad days, and I think I was my sore throat was getting better when I actually had the antibody done. Because that was one thing, is that you can't just walk in and get the antibody. It's appointments, and they're only doing it, at least around here, at the local hospital 2 days a week. And then there's another hospital, which is another 40 minutes drive, where they would do an additional day in the week. So we could get it Monday, Wednesday, or Friday, and I got it on the Wednesday. So I assume it helped, but I honestly can't say any more than that.
Yeah, and I know your voice, you were sick, seemed like you were very sick for about a week, and then your voice, you lost your voice, and it's back, but it sounds like you're still kind of on the mend, and even probably healthy.
Even then, you felt better, but you still got tired really quickly. Oh. So I was back at work, but I'd hit like maybe 3:30, 4 o'clock in the afternoon. Boom, you're dead. Like, I need to take some time out.
Well, I thought that was just being over 50. Not that I'm there yet, but
no, over 50 isn't that bad, believe me.
Yeah. We can move on. That was really. I hope everybody, that first story is a good one. Again, we really think the vaccines have done a lot. But the next item here is from Inspect. Is it Inspect IR, which we talked about, right?
I would call them Inspector Systems.
Oh, Inspector Systems. Yeah. See, you're much smarter than I am this morning.
But I like this because it's actually mass spectrometry contributing. And it's Inspector Systems COVID-19 breathalyzer test wins an FDA nod. And I'm always interested in what different testing approaches there are. And honestly, this week, I've been out visiting customers, and you still on-site visits. You either get they'll do questionnaires beforehand, or they'll ask to see your vaccination record before they let you on site. And some still test, which I'm honestly perfectly happy to do. So yesterday was a swab round the nose. So it's still in that kind of mode. So it's really nice to know does these other things. And this is Inspector Systems have a miniature mass spectrometer. That they basically set it up. The way they do the COVID test is that they're looking for exhaled chemical compounds that come with infection, and they've keyed in on 5 compounds that if you're infected, you're going to be breathing those out, and then the mass spec basically is looking for those 5 volatile organic compounds, and it's they basically say in terms of negative testing, they have a really high success rate. They also this is emergency use authorization. So between them and the FDA, they're saying if you have a positive test, confirm it with a PCR test. But they examined more than 2,400 people with or without symptoms, and the study demonstrated a false negative rate of less than 1%, according to the agency. And it's another nice thing about this: it's a 3-minute test. So it's it's relatively rapid, and obviously much less obtrusive than sticking the swab up your nose. So, as someone who has done mass spectrometry for years, it's really nice to see. Not surprising that it's contributing, but it's really nice to see how it's contributing to deal with COVID, or at least test.
Yeah, there's a lot in this article, and we got a little bit of limited time because we have a special guest here this week. But the fact that they shrunk the mass spec down to a suitcase in and of itself is something that's got to pique your interest, John, right? Like, what was the size of the first mass spec you used? It was
the size of the first one. No, the size of the first one was maybe a half a room size.
Yeah.
But when I postdoc to North Carolina, there was a big lab there, and it basically, I would say, your average living room would have the one of those mass spectrometers would have taken up the entire room. They were massive.
We had some archive ones that, like, we used to have them, like KCAS was like a museum at one point. We had them in our halls. I don't know if they were still there when you first started. Oh, they were huge. And now suitcase size is amazing. And then, the 3 minutes is interesting because that's not too long, but it wouldn't allow you to walk into an arena, right? That's where some of this goes. Like you go to a sporting event or a concert or something, but it's on its way for sure. And I wonder if, airlines and things like that, is 3 minutes seems worth it. But certainly for entering into, clients that we go to see and stuff, if we could take a 3-minute test, that would be fantastic.
Just on the miniature, just first of all, on the test, it's designed to be used under the supervision of a healthcare professional in doctors' offices, hospitals, and mobile testing. But you're right. I think it could be extended beyond that. And in terms of miniaturization of mass spec, that's been a big emphasis for years, because it is that convenience, looking to have testing at source of care. So, I mean, we had when time of flight instruments really kicked off around. When did I start with Kratos? That would be around 92. And those instruments, although they were still big, the Kratos time of flight, the company I worked for, could be lifted by 2 people. But we used to drive it around in a car all over the UK and Europe, and we'd pump it down overnight and then run people's samples. At the same time, Thermo had launched a small time of flight. And I'm not sure that was quite as convenient, or whatever. It was still small. But then, in terms of these volatiles, the miniaturization, because where the key is, I think the electronics have got much smaller, and then the turbo pumps have got that we're using to pump down, because the pumping technology for mass specs, because you're operating a high vacuum, the turbo pumps have got smaller and efficient, and so that's how they've managed to really shrink everything down. So the company I worked for before KCAS, Advian, we actually had 2 businesses under one roof. One was a CRO, and the other piece was actually started off looking at a nano flow source for low flow LCMS. Or infusions, but as they branched out from it, they've launched a miniature mass spectrometer as well. I'm not sure what size it is. I've never actually seen it, but there are a number of them out in the market. So, like I say, it's from my perspective, it's not often we touch on my realm. It's usually your realm.
Well, I won't go into how they tried to put flow cytometers in sub-Saharan Africa and drive them around in SUVs, and maybe Jeff's aware of something called a guava system they tried to do this with, and some other flows. But the problem was it couldn't get clean water, and even trying to get a flow cytometer put somewhere, no one knew how to operate it. It did not work. And now they're more keen in getting the sample out of there than trying to put instruments in there.
Yeah.
So, but we can move on. I think it's a great story. To that system there, we, over time will probably get more widely used, and certainly be, if we have more waves of COVID or future pandemics, it would be a great thing to be able to have somewhere where you could just, wherever you're walking into, you take a breathalyzer test and you can go in, right? That would
Yeah,
be.
I think, if people don't like the swab up the nose and sort of think, I'll hold off testing because I really don't want to do that, this makes it much, much easier. So
I think so.
hopefully it would encourage people more.
I wonder about price restrictions in our lifetime, though, might be part of it. I wonder how expensive that machine might be.
I would think it's probably. I'm talking off the top of my head, but I think it's tens of thousands as opposed to. Well, it's not going to be hugely expensive.
Yeah, not too bad. That's not too bad.
I don't know about the designer, but I would suspect it's just a single quadrupole and detector. So it shouldn't be. And that was certainly the Advian instrument was a single quad. Again, with looking at. I think their approach was more universities where you could get scientists walk up with a compound they prepared and just immediately run it into the mass spec and get an answer whether it worked or not. It's that kind of thing. So I think from our perspective, when we think of scientific instrumentation, they might not be that expensive. But
not bad.
The price.
All right. Well, we got one more COVID story, John. I think we can do this one pretty quickly. Why don't you tell us about, is it Dr. Crumley who gets infected? Good story here, too.
We'll give the headline. It's another rare virus puzzle. They got sick, got treated, got COVID again. And so far, and I'll emphasize this is a rare instance, but it was in the Washington Post, and the first person he cites is a guy called Greg Crumley, who was a retired molecular biologist. And he developed a sore throat and congestion, took a rapid test, and then saw that thick line materialize on the testing. And I have to say, when we got our positive test, the speed that line formed is like there's absolutely no doubt that we've got it. But he has a similar history to us in that he'd been fully vaccinated. He'd fully vaccinated with booster. Once he actually had caught COVID, knew he tested positive the first time around, so he got the course of Paxlovid. So he obviously wasn't doing some of the drugs that we were doing. So he did the full course of those, and Paxlovid dealt with it. And so the expectation was that would be him, and he'd be set. But like say, in a relatively small number of patients, they're actually seeing this rebound that once they finish the course of the Paxlovid, they see a resurgence of COVID symptoms. And I think the analysis is it's not a reinfection. They really think it's a resurgence of the virus, and so they're still trying to figure out why this is happening. There's some question: is the 5-day course long enough? Should it be longer? It's one of those puzzles that COVID has thrown out yet another curveball that we have to solve, and I'm sure they will. It's just it's there, and be aware of it.
I think there's latency maybe is involved here. We don't. No one really talks about if there's what the latency of COVID is, and what I mean by that is it kind of integrates and hides in your body, and then when you're stressed or whatever, it kind of can manifest itself. It hasn't been talked about too much with COVID. Certainly something to be determined, but it is, man, I don't know what these individuals are doing where maybe you too, John, you guys keep getting sick.
This is my first, this is my first time. And
I guess you're not too bad, but this Crumley guy, I wonder what he's doing.
Well, it's difficult when you're in a big city. How can you can't really, you're going to come in contact with so many people. That's the thing. You're sort of trying to get your life back to semi-normal, and so you just come in contact with people, and if you're unlucky, you get infected. I do want to emphasize there's a Pfizer study. So Pfizer obviously are aware of this, and they've gone back to look at it, and they tested. They looked at data from people who'd taken Paxlovid and placebo, and they actually saw that both groups showed this kick up after a few days. So that like I say, we don't really know why this is happening, what's causing it. It may not be Paxlovid related. It's just it's heightened awareness of it, and I'm sure we'll hear more as this continues. But it's just an interesting story to touch on.
Yeah, no, I think it's whether you're and then there's on the flip side there are still what's the vaccination rate in the country? 63% or something. It's kind of stayed at that level, right? So
I think that's the thing. It's relatively stalled.
Yeah, it's definitely it's not above 65%, I think, for the country, and so and got a massive population out there that's not vaccinated. That no, it's hard to say what those numbers are. I even have some family members that are not vaccinated.
And the issue is we've got variants that are coming through that are behaving differently again. So even, I'm not scaremongering here, but there's concern how even those of us who are vaccinated to how well the vaccine's dealing with those newer variants too.
Yeah.
So
it's something you're not. I think you said that well. Scaremongering is the word. It's just you being realistic. We don't know what's coming there. One could make a case maybe some of this has been around and we know that there's been COVID outbreaks before. I just wonder if we're just, going to have to kind of, I don't want to say just live with it. That sounds cold, but we certainly don't want to be back in lockdowns and going months at a time in our house and
I think
washing off our mail and stuff. I mean, we went kind of crazy for a little while. At least I know I was like,
And I think honestly that it was in the early days, it was right to control what you did and contact and things like that. I think we do have tools to deal with it. So it comes back to the first story. If we are vaccinated, you greatly enhance your chances of dealing well with it, even if you're infected again. Like I said, I didn't have a great week or two, but I came through it without having to even consider going to a hospital. And I think that for me, the key on that article earlier is the reduction in hospitalization rates is huge, simply because it means that not only are there less hospitalizations for COVID, it means that hospitals can turn their attention more to the other conditions they were unable to get to because of all these COVID patients.
Yeah, and you're seeing a lot more of that too. Like that's certainly many universities are starting to see other diseases like strep throat's coming back and influenza, all sorts of things like that, rather than just all being COVID, which is, that's a part of a sign we're starting to mingle together. John, we're running a little long.
Well, I tell you what. Well, let's do the next one because I think this is amazing, and maybe we could drop the one after that.
Yeah, you want to talk about SCIDs? Is that SIDS rather?
SIDS, yeah.
Very. This is a. Please go ahead.
Let's drop because if we're doing another one soon, we'll do the ALS one next time. I think that's fine. So let's get back on track. So the one non-COVID story we want to cover, this to me is, of all the things I read in the last however many weeks, this is just huge. It's like study finds link to SIDS for possible infant screening test, and this is a group in Australia. Oh, so SIDS to outline it is sudden infant death syndrome, and it pops up in the media every so often because it's unexplained baby deaths. It's babies who seemed healthy the next morning are just dead in their crib, and it's a big deal obviously for parents because they're like, what could we have done anything better? Obviously there's a lot of suspicion on parents when it's their kid it happens to. And it's actually, there's a group of researchers in Australia led by Carmel Harrington, who's an honorary fellow at the Children's Hospital at Westmead in Sydney. She actually lost her own child to SIDS 29 years ago, but this group were looking into it. And they actually identified that the nervous system enzyme butyrylcholinesterase is a strong marker of risk. It actually plays an active role in the brain's ability to wake up the body from sleep in response to difficulty breathing or another stimulus. So they looked at 700 dried blood samples taken from newborns at birth, and the researchers found that the levels of this enzyme were significantly lower in babies who later died from SIDS. so this deficiency could reduce an infant's ability to wake up and cry out if they're having trouble. So it's, I mean, I just thought this was because so this is just amazing that they found a chemical marker first of all, and B, it really helps answer those however many parents who've had to grapple with this to say this is not your fault, this is a natural thing, and hopefully now we can do something about it.
Yeah, I mean, you're right, John. It's it's to think about this. Carmel Harrington, 29 years ago, been researching it since to even discover the biomarker, this enzyme, is incredible science. But it's, as you touched on, this is oftentimes parents would be accused of things, or even like, I feel like I remember probably 20 years ago, it was like, oh, don't let your baby sleep on its stomach, don't let it
Yeah.
sleep on its back. I mean, it was just a lot of misnomers, and just, I think this really does put, I think it's really hard being a new parent, right?
Yeah.
Just one more thing to help maybe put people's mind at ease, and certainly something that they did it by dried blood spot, which is a wonderful technique, an easy thing to do, not hard to get a drop of blood and put it on a piece of paper. That's what this blood spot is, and then test it. So that's a real plus. They don't have to actually try to get blood from the child, meaning trying to draw blood, which is really, really challenging in pediatric studies, right? But it's a great story, and I think it seems like a strong marker. We'll see over time, but basically, if you lack this marker, that's bad, right? This enzyme allows you to wake up, and John, maybe it even has other sort of central nervous system sort of application over time as well.
Yes. So just in terms of numbers, I mean, it's basically, according to CDC, there's about 3,400 sudden unexpected infant deaths in the US each year, and it's SIDS and other things. And but although the number has dropped because of sleep advice and like you touched on, the don't let your child sleep on the stomach and don't have them overheat and things like that. But like you say, it's all about what can this do? This at least gives us a signpost as to how we can possibly address this. If you know from day one that the child is low in this enzyme, how can we affect that? And
knowledge is so powerful. Even if you know your child's in this category, that becomes something that they, maybe there's not an active treatment today, but over time there's going to be. And then also just the thought of, man, that would really freak you out if your child does test.
Sure.
So in this case, negative, right? That's if you lack the enzyme, that's when it's bad. Anyhow, great story there, John, and really good movement in the field of SIDS, which is something that I hope everybody's familiar with. It's a high number, 3,400.
And that's
seems like a lot.
And that's just in the US. I mean, obviously, it's that number will be matched worldwide. So just moving on to the final thing, and I think this is a quick topic, but interesting to note how things are changing. With COVID, when it first kicked in, obviously money really flowed big time into biotech and pharma companies, but obviously things go through cycles, and what we've seen, and we see it with the broader stock market, which is losing substantially at the moment. But I think there was concerns about there were so many companies out there in terms of biotechs that the industry was going to right size itself at some point, and that seems to be happening. So much so that Fierce Biotech, which is one of the newsletters that we check out on a regular basis, has started a layoff tracker to really look at which companies are laying off and why. And so they started from January, where there was 8 companies total had some layoffs, then February 5, March 17, April 17, and May 4 so far. And it's a variety of reasons. Some it's just to reduce costs and cash savings to hopefully prolong the life of the company. Some it's change of research focus. Some it's the FDA rejection of lead compounds. So that's part of the natural cycle. And then else, that's more coincidental. It does tie into failure of key drugs in pipeline, which is a similar thing. And then others, it's slimming down the operation while looking for bio. I thought it was interesting just to talk about how things have changed recently. So we'll see where it takes us. Although in the same breath, actually, there's been other articles out there that large pharma companies are awash with cash.
Yeah.
So at the moment, if they want to buy companies, they're in a really, really good position to do it. So I would expect to see more of that coming up in the future as well.
Yeah, some of this might just be a swell, as you kind of, I think of it as like it just the biotech field just got such an infusion in it over the last few years that there's got to be some regression. And then there's naturally things that happen, like a pipeline gets stalled, something's not working. All of that leads to a certain degree of like downsizing of companies, especially biotechs. But you touched on the real positive light, and I'm glad you did. That's pharma, large biotechs. They have money to burn, and there's a lot. They're spending it they're going to spend it, and more than likely it'll be infused into some of these areas that are trimming down, right? I mean, but it's a little concerning that Fierce Bio has now got a tracker, which is kind of that in of itself a sign. But I think when you look at some of these companies, like the size of them, it's not surprising when they hit a snag, right? Some of them, their phase 3 fails or something like that. That company is going to have a 25-40% of the staff laid off due to that.
Yeah, and there have been some big ones because of that.
And then like the other marker is like IPOs and stuff like that. And there's been some slowdown, but there's still periodically
the
numbers coming out too.
The numbers are much reduced from where they were, but you still see some coming through.
Yeah.
So, well, I mean, I thought it was interesting to highlight. We'll follow it obviously in coming months, and we'll see where it all goes.
It's just we don't want to be alarmist. There is some industry turnover here, and it's something we should keep an eye on. I think it's important to touch on. But maybe without further ado, we can kind of move on to our main portion. Of course, today we have a very special guest. Jeff Hester, who's our Senior Director of Cell and Gene Therapies, is here with us. And so today we're going to talk about just that, cell and gene therapies. And so, Jeff, I hope you're excited and ready. So where do you want to start, Jeff? Maybe give a quick little overview of what is cell and gene therapies, and then we got a whole list of sort of items and stuff we can go over. But welcome, Jeff, and maybe you can give us a little background on what are cell and gene therapies.
Gentlemen, what a pleasure to join you today. I've on the KCAS Weekly Bioanalysis podcast. I've been following you from the very beginning as I've worked with many colleagues here over the years from KCAS and KCAS being a local CRO that I've monitored and watched over the years, and very excited to join the company over the last year, but also join the podcast and your sage wisdom, camaraderie, banter, really mixing scientific topics with lighthearted discussion, as I think has put many of people's minds at ease over the last year or two with COVID. So, one, thank you for all that you've done and getting this our message out to the general population, but also to our clients in general. So, as Dom alluded to, I'm a senior director here for our cell and gene therapy program. We have invested heavily over the last year or so with both technologies and infrastructure to support our pharmaceutical clients so that they can move their life-saving therapies through the drug development pipeline and ultimately into the marketplace where it will help patients and save lives. So I come and lay my really infrastructure on top of a firm foundation that was already established here at KCAS in our cellular bioanalysis team. What I bring is nearly 2 decades or more, actually, of molecular and cellular bioanalysis experience, both in a CRO also as well as in a clinical marketplace. I started doing, if you look at the techniques that are commonly used for cell and gene therapy, and we can slice this topic many ways, but I'll start with where my background, really started back in the late 90s with real-time PCR, and that's a major technique that's used as really, and now of course it's now moved on to other techniques such as droplet digital sequencing, whether it be Sanger or next gen. And then, of course, I have my exposure over the last 2 decades and leading teams on the cellular bioanalysis, whether it be flow cytometry, ELISpot, or just biomarker readouts for front-end cellular assays. So my background really is diverse, and it fits uniquely with in the cell and gene therapy space. That there are many techniques that must be mastered. There are many techniques that must be offered in order to really help our pharmaceutical clients once again move those those drugs through their development pipeline. And KCAS is really provided and put together a premier suite of these technologies for our clients.
Yeah, great, Jeff. Thanks for those, first the kind words, and really thanks for that little overview. And we are excited. Obviously, we had some nice cellular work before you joined us, but as you said, you're bringing a wealth of experience, and of course, you're amassing what I'll call the Avenger team. It's the A team you're getting there. So, Jeff, we could take this many ways, but you kind of touched on it early on. I think some of our audience might be wondering, well, what are these cell and gene therapies? Of course, they're probably familiar with it. It came to the forefront with the vaccines, but maybe you want to tell us a little bit about like some of the, one that sticks out is CAR T cells, but there are other, when you look at the number of approval of cell and gene therapies, is about 27 of them. They fall into, a lot of them are in the hematopoietic stem cell space, which is that's a true cellular therapy. And then you have, traditional gene therapies. Then, of course, the combo of cell and gene therapies is that CAR T cell. Maybe just, I kind of gave an overview. You can take it wherever you want, but I think it would be nice to just touch on why it's so important and why is it coming to the forefront, right? Like
Sure, absolutely. It's I mean, I know we've been speaking as scientists, clinicians, physicians, medical personnel over the years. Personalized medicine. Anyone who's really been in this field, this in 80s, 90s, the day of personalized medicine is coming. And I think really the cell and gene therapy is really gets us to that point where we're actually looking at the individual cells, or we're looking at the individual's genetic information. We're modifying the cellular information. We're modifying the individual's genetic information. For, of course, the end result there is to provide life-saving cures to these individuals. So there are many ways, and it's a very diverse topic, and it's a very broad topic. So we kind of I'll go from an overview of it, and we can drill down into individual topics. So if you look at cell and gene therapy, you're looking at cellular therapies, you're looking at genetic therapies, and really cell therapies. They aim to treat diseases by restoring or altering certain sets of cells, or by using cells to carry a therapy through the body. Whereas gene therapy aims to treat diseases by replacing, inactivating, or introducing genes into cells inside the body in vivo, or outside the body ex vivo. So if you look at those are two broad topics, cell therapy and gene therapy. You can start slicing them up now. You can start slicing the cell therapy. If you look at that, you have non-modified cell therapy, which many of us. I spent well over a decade really working for the transplant community, providing novel diagnostics and helping therapies in that space. And there you had a lot of you had a lot of autologous therapies. So you have non-modified, just putting cells back into the body, very specific subsets of cells back into the individual, the patient. You can look at gene-modified cell therapy. So that there you're actually modifying. Dom alluded to one of those is CAR T cells, which is very very prevalent now with regards to investment in the biopharmaceutical community. And then of course you split off a little bit on a little bit is you talked about gene modified cell therapies. Now you talk about just gene therapies, which is the introduction of the payload through viral or non-viral methods into the patient. And then I just alluded to another. How do you split those? Now you've got the viral versus non-viral methods for introducing that payload into the individual. So it's a very broad topic, which is why the techniques that are really, I'll call it customized for this, what we use to help our pharmaceutical clients, it is diverse. It is very diverse, and it's the breadth of expertise needed. Dom alluded to, you have to have a deep knowledge of virology, microbiology, cellular immunology, all of those are keys to really serving our clients, because this is a very complex topic.
Yeah, no, you're. I think it's great. We're digging in a little bit. You said some wonderful stuff in there around like kind of the evolution, right? It was autologous therapies, self therapies, self cells, and now it's more cells off the shelf, and now it's autologous therapies with gene mutations, and it's going to be like cells off the shelf is how I think of that. Cells in a bottle that are going to have gene therapies, and it's wonderful. The treatment range is broad, but I think we can, like, part of what the goal of today's kind of main agenda here, Jeff, is you started to talk about the platforms and really that, like, the tools that are used, the talent you need, and then really the biggest challenge, and we'll talk more about that, is what does the PK profile look like, right? That's a tough challenge, but maybe just drill into what are some of the tools and to use in the cell and gene therapy space, and then we can even dig in about how KCS is solving some of them. But let's jump in on and maybe talk about how do you go about tackling a cell and gene therapy pharmacodynamic PK assay?
So, the therapeutic itself is either a cell-based product. So, if you're looking at, say, a PK, and sometimes it looks a lot like a PD for cell and gene therapy. But if you're monitoring the therapy and looking at distribution studies or integration of these various products, these cellular and/or genetic products, this with regards to cellular, you're looking at monitoring those cells. So looking at the distribution of those cells, those modified cells, such as CAR T cells or similar, in various tissues, in circulation. So you're using the techniques used for that in many regards are flow cytometry, proliferation studies. You're looking at some sort of cellular bioanalysis technique that allows you to quantify those cells in some manner or characterize those cells in some manner using unique epitopes specific to the CAR T cells. Also, you can look at the cells themselves, but you can also look at the genetically modified information within, or the genetic information within those cells. So that itself can be a way of quantifying that therapy within the host, whether it be an animal or human. So there are many different ways to look at that. You have to, just like you do for any other PK assessment, you look at the drug. How do we identify and then quantify that, and then monitor it throughout the time course study? And for us, it's it takes relatively complex techniques, like it does for many. If it's cellular, we use flow and/or genetic techniques. So it really depends. It's customized. It's a customized solution for our pharmaceutical clients. So in a roundabout way, you got me there, Dom. It is very much customized to the therapy itself.
Well, no, and the Ellie spot's another tool for that, right? So it's not, so part of it is the biggest challenge here is, if you think of a traditional PK, you get a reference standard somewhere, some manufacturing GMP certificate of analysis tells you what's in that, how much it is, what's the purity of it. When you go to tackle a PD, I think of it more as pharmacodynamic. You have none of that, right? So then clients frequently come to us and they think, they're conceptually, it's here's what we think we need. And it could be a flow-based method. It could be an ELISpot method. It could be a genetic, some sort of PCR or even digital draw PCR or even next gen. I think that the part that really the value we bring, Jeff, and this is more specific to KCS, is the ability to take what the client is thinking of as and they might do it in their laboratory, right? And then it becomes, how do you do it across a clinical trial? That's really where our expertise lies. And Jeff, I think that's part of what you've been doing for decades. And that really becomes like what we're amassing, right? That's what we're trying to put together. It's the platforms, the depth of the team, but it all translates into really getting people to get over that hurdle of just looking at one laboratory doing something that's then going to have to be able to go from a single lab doing it more in vitro to a single clinical site to 10 clinical sites. And that's really, I think, where we bring a lot of value. So maybe I don't know if there was a question there for you, Jeff, if you want to add to it, but there's a lot. That's really, because running these techniques is in and of itself an art. It's challenging. But that's where from taking it to and being able to support like a single site, 5 sites, that's really, it's the sample logistics, all of that becomes. And then of course, it's not just, oh, hey, here's a PK profile. It's oftentimes multiple facets of pharmacodynamic testing, functional testing that allows you to say, okay, this did go into the individual. It's having some it's we're proving our hypothesis. The mechanism of action is working through this sort of functional cellular test or through this genomic test, right?
Absolutely, Dom. You really alluded to something that's near and dear to my heart. It's, the holistic approach to what we do as a CRO, which is really customized solutions, not only for the science, but really a customized solution for their entire program, uh, for their trial itself. Nothing that I do will make any difference, in the, if you will, in my team in the lab, if the samples don't arrive here in a high quality state. So that's, like you said, this logistics. It's being a partner with our clients, being a partner with the clinical sites, helping train them, and we do that all the time. I work directly with clinical sites to make sure that work instructions are clear, that they can get us the samples. And for us, most of our samples don't come frozen on dry ice. We're dealing with live cells in many regards. So when you talk about the cellular component, flow cytometry, most of those are live cell assays, or recently live cells. Sometimes they can be stabilized, but in many regards, we're getting live cells here. So they have to get here quickly. They have to get here viable, and I have to get them in a high quality state so that I can give them an answer, data that makes sense for their trial. So
So
that's,
Jeff, can I quickly jump in? When you
of course.
say quick, when you say quickly, because obviously with LCMS we're always talking frozen samples, and we don't have to worry about this kind of logistics. What sort of time frames are you talking to get
Sure.
get things to you?
Sure. So, we work here at KCAS with, of course, with the large couriers that will get us samples within 24 hours of blood draw. And that's throughout the U.S. And so, routinely we have staff waiting here early in the morning for that early delivery, so that we can start the cellular processing bioanalysis within 24 hours or less of the blood draw itself at the clinical site. So that's to Dom's point, that's one of the great things about this is it's the service. It's part of the service that KCAS provides, with weekend having people and staff, qualified staff here, for a Friday draw, Saturday analysis. So John, that's a great question. It's I'd love to have samples here, you know, if you will, immediately after draw, but of course we have to have them sent to us, and uh, as a low, many of our clinical sites are throughout the country and not local. So
And can I'll ask one more follow up. So as a complete neophyte to this, what's the issues if you're going beyond that 24-hour window? Is it death of cells, or what's the problems that you then start encountering?
yeah. So, every assay, so I use 24 hours. That's just happens to be top of mind today because that's what, we're working in. So we do a formal stability study for every cellular assay, as we do for many assays, not just cellular, but for us, stability assays are very important. So we do we try to see if we have 24, 48, 72, hopefully in stabilized cells, we can get out to a week or two. And there are some assays that allow us to do that. And to your point, so 24 for certain assays. Really, you're looking at protein synthesis epitopes that are expressed or transient. Some will be, stability can be caused by these specific epitopes sloughing off. So it many different things, but really it's not this. I'll call it the very specific cellular epitopes that are unique, that need to be monitored for their therapies, not the major, I'll call it immunophenotyping epitopes. Those are pretty stable for a more extended period of time.
Okay.
So really it's the very specific ones that are unique for that drug, that therapy. So
Yeah, maybe I think it's a great place to kind of drill in even more, right? So part of the instability of samples is non-specific activation of cells, right? So if you have a whole blood sample, John, your question's a good one. It can be a combination of a false positive or a false negative. And as Jeff sort of indicated, there are like immunophenotyping, a traditional what we call a T cell, B cell, NK panel. That's an HIV panel. That's actually stable for like almost 72 hours. Well, really, Jeff and I would probably tell you 48, but I think the actual guidelines say it's stable for up to 72 hours. But those are hardy immunoglobulin-like structures on cells, and so they stay pretty stable. The real meat of it is when you look for rarer populations that have markers that are their size and structure create the instability. The density of them make them a little bit harder, meaning they're lower density, and then the functionality of it is lost, and that can become a real falsely high or loss of a given functional test. So all of that plays into, some tests require within 4 hours, and that becomes really, really hard to do. Obviously, you need a clinic, which was local to us that we could do studies like that with. And then, of course, I think, and Jeff and I'll circle it back to you. This is all done during feasibility. That's one of our strengths, right, Jeff? Everything we're talking
Absolutely.
about, we work backwards from, and we leverage our experience from it. We start with, hey, what's your goal here? And then we help our clients get there. And one of the biggest things we're learning, Jeff, is 24 hours of stability is just outside the window. You kind of need 30. And
Yeah,
maybe you could tell us more about that. Go ahead.
sure. Yeah, no, I mean, it's really a shoulder to shoulder approach with our client. We're heading into this together. And yeah, as Dom alluded to, all of this, I can safely say a certain number of hours only because we did an extensive amount of feasibility work up front, and really, both in pre-val and val for these assays. So we know what that outside time point is that we need to stay within. And every assay is, once again, it is truly custom for every assay. So generalities, yeah, they can be used. Usually this works. Usually we're stable, but really the data tells you that. And then of course that's what that's our guardrails. We try to stay. Our goal is to stay within those guardrails and help our clients stay within those guardrails. So yeah, absolutely. So it's I would think that the best way to describe what we do is it is active management of a project, and that is not just of course, the technical teams. I alluded to it, the logistics. That's your really your teams that provide the kits. That's the teams that are the program managers. That's really senior management, all being aligned to make sure that we can provide that customized solution to our clients. And for cellular assays, it is no trivial task, and some CROs do it well, and others just don't have the infrastructure and the focus to do it well. And KCAS is one of those that I feel does it very well and has a focus on that. And that's back to my original comment. We have, especially since I joined, but even before that with the foundation, we have a focus on becoming really a premier laboratory for cellular and now molecular bioanalysis in this market.
Yeah, and it piles on. It just our legacy, our 40+ year history,
Mm-hmm.
our ability to provide solutions in terms of logistics has been. That's why we have a clinical supply and kitting department. It started from stabilizing metabolites and small molecules, then it got into biologics, and then it got into CPT tubes, PAX genes tubes, and Cytochex BCT tubes. Right, Jeff? Before we even came, we were offering those services, and all those things through solutions. I think the biggest challenge for you, Jeff, sometimes you got to be the bearer of bad news, right? And I hope you know what I mean by that. You got to go to somebody, and this is very hard to do, Jeff, because you have to go and say, hey, look, we're gonna save you a lot of time, money, and effort during this feasibility, saying, hey, this isn't gonna work. And that's hard, right? And then then it's like, okay, well, maybe you can't get this specific, I'm gonna say regulatory, often we find, or this particular receptor for a regulatory T cells or central memory or whatever they might be looking for. And then you got to start looking at alternatives, right, Jeff? That's the power we bring.
Yeah, and that's, as scientists, we will do anything and everything to try to really find that solution. That here's what we want from the client. let us do our thing to find that solution for you. And like Dom said, sometimes even with all of the, I mean, you start adding up the years of experience you have within a team, and you start to get into literally, 100 years or more, of experience in many different areas, and deep cellular immunology backgrounds, with our scientists, and every once in a while, you ultimately have to be the bearer of that bad news. You have to admit it at some point that we can't provide a solution that fits specifically what you're asking to do. But
Yeah.
here's what we can do, and here's what we'll do to make this work. And there are many unique situations where we absolutely go a little bit outside of, I'll call it, traditional CRO work, mainly because of our dedication to the client. And we want to help them and guide them, and our years of experience is valuable. And they, I think that's to what Dom's alluding to, is that is empirically derived experience. That is something you have to generate the data, if you will, in-house with your people in your facility with your teams, in order to provide that really that expertise. It's something that you can't just wave hands when you're looking at logistics. You've got to say, I know we can do this, and here's how you do it. And that's like you said, 40 years of experience here.
And Jeff, it's actually I think it's very powerful to be able to help, even if you have to be the bearer of the bad news. Because what's the flip side? And you've experienced that, meaning another laboratory has shown that, oh, these samples and this will work, and then they invest in qualifying or validating that method, and then they do a clinical site study that might have maybe 5, 10 sites. And then what happens, Jeff? They test the first quarter of those samples and they get no data at all, right?
Yeah,
and then they come to us and they say, hey, can you help this? And there's two real major things that happen. It's the integrity of the sample, and then that panel was flawed from design, right? That I think is unfortunate, and it's not malicious, right? And we're not saying those people over there that those particular labs weren't good and this and that. But what we're saying is, we tactically, before we're going to start drawing a clinical trial, we want to make sure it's going to work, right, Jeff? That's really the power. And then before you start drawing, you can start to say, hey, let's alter your hypothesis to fit what we can stabilize or what can be produced, right?
And that commonly happens. I mean, that's really the sage advice that you're providing. Your experienced advice, the advice that comes from decades of late night hard work that you can provide. And you're right. No, I'd like to, and I truly believe that no scientist got into this to be malicious in any way. And what we do is we're always trying to help. We're overachievers. Scientists by nature are overachievers. We try to please. We try to satisfy. We love troubleshooting. We love the art, if you will, of all of this. So when a solution is not found, or it's a lot of times it's naivete. I mean, honestly, we've never done that before, so we gave it our best shot, and then we get those projects from those labs. It's never malicious. It's
or even subtle, right? Just a misgating, like, oh, we think this population's here, and then the reality is it's just it's kind of like these are really sophisticated pieces of equipment that require quite a bit of knowledge, and then we can just go in and it's like, that's probably not right, and it's hard to say that, right? But it's still very powerful and palpable, and that I want to emphasize to our audience that is really what we're about. We're not going to try to like sugarcoat it. I mean, sometimes it is like, hey, this isn't working, and that's where really, Jeff, we've been down that path together, but it's also I want to emphasize that it would be much worse if we do it well before a patient's enrolled versus a year later, 2 years later.
Absolutely.
And somebody's looking for answers. The drug kind of works. We need this pharmacodynamic marker to help us stratify our data, and you're sitting there going, well, we're not sure, right? So it's wonderful stuff. Yeah.
That's would be an uncomfortable position to be in, and we're all proud of the data, and really goes back to that one word, partnership between us.
And then, Jeff, really the knowledge you and your team bring is that controlling of the experiment, and truly, like, it's not as simple as just like one set of QCs. It's much more than that. And without, like, I don't know, we don't need to get too much in detail on some of that technical stuff, but like, that's really where I think when you talk about the hundreds of years that we have, That's what separates us, right? It's that ability
Absolutely.
to say, hey, with conviction, before we even draw a sample, this tube of blood can be stabilized for 30 hours or 24. It only gets 18, or maybe it gets 4 days, whatever it is. Then you work backwards. And really, we can think of a few occasions, as even the last few months, Jeff, where we've helped people alter the way they're designing their trials so that they're set up for success, right? That's really what
Absolutely.
it's about.
Yeah, it
Um,
really is. I mean, it's being upfront with them, and those early conversations you have when you're and as Dom alluded to, we get projects that we're rescuing basically before they either a failed initial trial, um, but where we get in and we look at old reports, old development reports, and we provide our scientific guidance on that there's a reason why it didn't work last time, and here's what we would
yeah.
change, and then. And that is extremely valuable. And no one here wants to see anyone waste money, especially when we're talking about bringing bringing life-saving therapeutics to the marketplace. We're all invested in this.
Yeah,
We're
and it's a patient sample. It's not like a television where you can just make it again, right?
it
That's the
is,
part that it becomes so precious.
and
And then
that's
I think,
yeah. Sorry, that's
no,
the
no,
and that's really the Dominic. That's why the logistics, and that's why knowing you have to know what is required to do these type of cellular bioanalysis, because that time point, once it's drawn, can never be gotten
mm,
again. You
yeah,
never get it again. And
yeah,
that's why when we see a sample is being shipped, we'll
yeah,
do whatever it takes to
yeah,
analyze it when it comes in, because
relentlessly. Yeah,
relentless, because that time point will never be drawn again.
yeah. Whether
And
it's
that's
Christmas Eve or Thanksgiving, Jeff, right? That all happened
absolutely.
this year. Yep, where that we have a sense of urgency about us that must it just that's why we got in this. That's why we come into work. And Jeff, some things that can really impact it is just even the anticoagulants, small things that we see sometimes where you look at it and you can immediately think, oh, they shouldn't be collecting this in K2-EDTA, or oh wow, they're using sodium heparin, right, Jeff? Immediately you can really think like they just grabbed what they had sometimes, right? Instead of, we may actually do studies to look at that. That's just scratching the surface in terms of
Yeah.
how we can solve problems for people. So we're this has been a great conversation, Jeff. We're kind of getting on in terms of time. Maybe if you want to give us some parting thoughts, we can wrap up. But it's been a wonderful discussion. Certainly over time, maybe we can get you back. We'll maybe drill in specifically to something. But it's been really a pleasure to talk to you here, or at least kind of share what cell and gene therapies are, how we do them at KCS. Any last parting thoughts before we put you on the hot seat?
Yeah, no, I appreciate it. It's been a pleasure, and I think Dom, we'll definitely go deeper in many different topics, and especially maybe in once again in specialized or more focused areas. This is more of a general overview, of course. I think the one last thing, and I'm as someone who's really started here only within the last year, and what I've seen that was has been here over the last 40 years, and the expertise in other departments that really are customized. It could be me, it could be someone else in LBA, it could be in someone else in Biomarker, it could be someone else in LCMS. They'll say the same thing. This is about customized solutions for our pharmaceutical clients.
Yeah.
So we work together as a team, because we didn't allude to it here, but all of these cell and gene therapies require techniques outside of what I provide in my group, whether it be in biomarkers, whether it be in the LCMS group, whether it be in the ADA group, the immunogenicity group. It is a full spectrum of bioanalysis techniques that we use here at KCAS to once again, it goes beyond me, and my colleagues are experts in their field, just like I feel like our team has that expertise. So it really is. It goes beyond me, and I'm very thankful to have the opportunity to work with so many dedicated scientific experts that make my cell and gene therapy program successful.
Yeah, no, and you're making me want to do push-ups, Jeff, or jumping jacks or something. You got me all fired up because that's really what it's about. It's not just Jeff Hester's cell and gene therapy group, and that's a powerful. But we've got 5 other senior directors that bring a whole world of platforms and expertise to help in this space as well. Not just cell and gene therapy, just like your platforms can help small molecules and large molecules. But that's just part of what makes our us, I think, unique, separates us from our competition.
Yeah,
It's all under one roof. It's all of us can, orthogonal approaches can happen very rapidly and very quickly at KCS because, again, it's all under one roof. all kind of in sync together. We're all rowing the same way. So Jeff, we're running short of time. We're even going to cut down the hot seat questions. We're just going to ask you two. Tell us your favorite dish to make.
I think my favorite dish to make, that's an easy one. So I love Indian cuisine. So I've spent the last couple decades, I think honing my craft. At least my family would say so. Of
Nice.
chicken tikka masala, chicken tikka saag, shahi korma. Many different curry-based, but also vegetable-based. Dishes.
Yeah, well, that's wonderful. My wife would love to come over anytime. It's her favorite. I can make something in an Instapot, but I wouldn't call it. It's not too good. I'm sure you don't use an Instapot.
We do not. That's usually
now you got me thinking again about ordering Indian takeout this
Yeah.
week.
I've gotten relatively good at making like garlic naan, even homemade naan and stuff. So it's it depends on the night. Sometimes you buy it and that's some good quality products, but sometimes you want to spend the time and try to make it at home. And but it's usually a weekend type thing, and then you make 4 or 5 dishes, and then that goes for the family for the week. So yeah,
I could only imagine how sweet it is to make your own naan, because I do love a good naan, and that's impressive that you make that. And it's unfortunate that we are a little short of time, but we got to ask the second one. What's your favorite adult beverage?
Oh, sure. Yeah, that's a good one. I think it's easy to say. We're heading into some warm weather here. For me, it's a freshly muddled Mojito, at home. Just enjoying that with maybe a little Cuban music, enjoying that on the deck or in the yard.
There you go. I like something called the 007 this time of year. It's Stoli orange vodka, ginger, 7-Up, and a splash of OJ. 007.
Sounds
good
refreshing.
There.
Sounds refreshing.
Well,
for me, it's like
go ahead.
For me, it's kickoff of G&T season. So yeah, we're back on the gin and tonic.
That was my second choice.
Check
definitely.
out. You want a good gin? Get Quarantine Gin. It's a good one. They made it during COVID. It's a nice price point too. It's called.
I'm still going to always tout the Chase.
Yeah.
Chase gin, the extra dry, and they're now available in the US. I'm a happy man now. I don't have to rely on duty free. So.
Yeah, no, again, Jeff, it's been a pleasure. We'll look to try to get you back in maybe sometime in the fall, a couple months from now, after you get situated in the move. We didn't even talk about the move. I'm sure it's going well.
Yeah, because
we'll get you back in.
We do want to talk about your lab setup, etc.
Yes.
So that'd be a good thing to talk
and
about.
you can. It's a Cadillac, right? You must be psyched to have
custom built facility just for what we're doing. Unidirectional workflow for molecular, dedicated rooms. What a wonderful opportunity to set up a first class, leading premier laboratory here.
All right. Well, the producer's giving me the timeout sign, so we've got to run. But Jeff, thanks. It's been a pleasure, and we'll catch you all on the funny pages.
Yep. Thanks very much, Jeff. This was great.
Thank you, guys. Wonderful.